India, and today I'll be talking about how three naturally occurring polymorphisms within a single gene interact in a non-additive way to affect life history adaptation in the wild in Drosophila melanogaster. So epistasis fundamentally affects the translation from genotype to phenotype in a non-additive manner. And in this case, the phenotype of interest is fitness. And so we're familiar looking at how you can move along a genomic space and how different allelic combinations can increase fitness. But what is fitness? It's composed of different traits that aren't necessarily correlated. So it's perhaps more appropriate to think of a more complex adaptive landscape with multiple peaks and valleys. And when we're looking at a clonally reproducing asexual organism, you can very elegantly move from an ancestral genotype and trace as mutations arise, fix, and increase fitness. But when you're looking at a diploid organism which reproduces sexually, mutations may not fix in a population, and so you're going to have all sorts of allelic combinations segregating in a population. An additional level of complexity is that the entire fitness landscape depends on the environment. So it's context dependent, and a trait that increases fitness in one environment may not have the same positive effect in a different environment. And when we're looking at adaptation, we commonly use environmental gradients to look at the association between genotype, phenotype, and the environment to infer the adaptive process. And so when we're looking along a latitudinal cline such as this, there are common adaptive patterns that we see. In particular, there's an increase in somatic maintenance, so increase in stress resistance and body size in northern environments, whereas there's an increase in reproductive output in southern environments. And so what I'm talking about today is how you would have non-additive interactions among different alleles that directly relate to fitness in these different environments. Specifically, how you could have one genotype, big A, big B, big C, that increases fitness in one environment, whereas the alternative allelic combination, little a, little b, little c, can increase fitness in a different environment. I'm going to look at this using Drosophila melanogaster. An important trait in their adaptation to northern environments is a reproductive diapause. And this is a reproductive quiescence as an adult, and so essentially the females stop reproducing. And you can see in these dissected ovaries that females that are in diapause, this pair at the top, the eggs have stopped reproduction, or the females have stopped developing the eggs at a certain stage. Whereas this set of ovaries that are not in diapause are the eggs are fully vitellogenic and they're ready to be laid. And so this again encapsulates this trade-off between somatic maintenance and reproduction. The gene couch potato has been shown to be involved in the propensity to diapause, both in Drosophila as well as other dipterans. And in Drosophila melanogaster, the level of couch potato expression is indirectly related to the frequency to go into diapause. So that means that in an allelic series, a hypomorph, lower level expression, has a higher propensity to diapause. Couch potato is a putative RNA binding protein. It's involved in transcriptional regulation, and it's expressed highly, although not exclusively, in the peripheral nervous system. In addition to being involved in diapause, it's been shown to be involved in other life history traits. And so these things together make it a very good candidate gene that's sensing and responding to environmental change. Work in the Schmidt lab has previously mapped a SNP in the couch potato gene which changes the propensity to diapause. Both the diapause phenotype and the SNP are clinal on the east coast of the US. So there's an allele that occurs in the south that has a low propensity to diapause, and an allele that occurs higher frequency in the north that has a higher propensity to diapause. And so in an independent screen, in collaboration with Bergland and Dmitri Petrov, we resampled the North American cline. And this was done about 10 years after, so this represents an independent sampling of the cline. We identified 3 SNPs of interest in the couch potato gene that are clinal. This third SNP that I'll refer to as SNP3 is the same SNP that was previously identified. So that's an independent validation of that SNP. The other two SNPs are both upstream, and they appear to be intronic. And as I said, these are all clinal, so they all have one allele that occurs more frequently in the south and one allele that occurs more frequently in the north. And what I'm interested in looking at today is how there can be non-additive interactions between the southern combination of alleles, which will be shown in red, or between the northern combination of alleles, which will be shown in blue. So to begin, I've asked if these allelic combinations occur more frequently together than you would expect. And to do this, I used a population that was sampled close to here in Raleigh, North Carolina, and I created an expectation of what the haplotype distribution would be based on the frequency of each allele. I then compared that to the observed frequency of the haplotypes, and as you can see, there's a very big deviation with the southern and northern haplotype combination occurring more frequently than you would expect based on the individual alleles. And so this led to the hypothesis that there are non-additive interactions with respect to performance that lead to emergent fitness properties for the northern and southern haplotypes that are specific to adaptation in those different environments. And specifically, because the two SNPs are intronic, we think that any differences identified may be due to differential couch potato expression. So to test this, I used the Drosophila Genetics Reference Panel, which is a series of 192 infrared lines that are fully sequenced. All of the different haplotype combinations occur in these lines, but one occurred at too low frequency to use. So for the other 7 combinations, I built population cages that were consistent for the SNPs of interest. To do this, I started with 9 lines that were consistent for the 3 SNPs, and then I let them recombine for several generations so that they remained fixed for the alleles of interest but had a mixed background. I then ran them through a panel of traits that are thought to be important in life history adaptation. And what you can see here is that star indicates the significance of that specific SNP with a value of 0.05, and the color of the star indicates whether the southern or northern SNP had higher performance. But so you can see that all the traits were affected and that some traits were influenced by multiple SNPs. But I'm not as interested in how these SNPs independently function, but how they interact together. So to do this, I developed a null hypothesis of pure additivity. And so I'm going to walk you through the analysis using the starvation assay, which is pretty easy to understand. You see how long the flies survive without any food. And their survival time in hours is measured on the y-axis, and the different genotypes here are shown on the x-axis. So you can create this prediction of pure additivity based on the performance of each of the individual SNPs that compose the whole haplotype. And so I can do that for all the different combinations of alleles, and as you can see, the southern and northern combinations are expected to be somewhat intermediate. I then moved from the absolute survival time to the relative survival time. So the size of this sphere represents the proportionally longer that the flies survived without food. And then I moved to a 3-dimensional space. So each of these axes represents one of the SNPs, and when you move along them, you just change that single SNP. So you can look at the predicted phenotypic response by changing a single allele in the haplotype combination. And then I compared that to the realized phenotype. So this is the actual cages, what they're measured. And you can see that it's drastically different. I looked at the deviation between these. So a positive deviation means the flies are surviving longer than you would expect, and they're shown by a solid or filled-in shape. The negative deviation means the flies are surviving less long than you would expect, and that's shown by an outline shape. And so again, this is the deviation from the expectation for starvation. But we can look at that for all the different traits that were measured. And there's a lot going on in the slide, but I want to draw your attention to a few things. First of all of the big blue circles. These are non-additive interactions among the northern combination of alleles that's increasing the survival time more than you would expect for suites of traits that are associated with northern life history adaptation. So diapause, starvation, chill coma, body size, etc. In comparison, the southern haplotype has much higher reproductive output than you would expect based on the alleles that compose it. And so this encapsulates this classic trade-off between somatic maintenance and reproduction. Additionally, you can see that there's a lot going on with these purple circles that are the intermediate haplotypes, but there's no directional pattern occurring with these intermediate haplotypes. And as I said, we expected that these differences that we quantified may be due to difference in expression of couch potato. So this is initial qPCR results for the different haplotypes at a single adult stage. So you can see that the northern haplotype has a much higher delta CQ, which means it has much lower expression. And when we look at the deviation from the expectation, that's drastically lower than you would expect based on the individual SNPs that compose that haplotype. This again follows the expectation that we saw with the allelic series, that lower levels of couch potato expression increase diapause. And we would have expected that there was also a higher level of expression of the southern haplotype. We didn't pick that up with this single life stage, but I'm going to go back now and look at different life stages as well as trait-specific expression, and I expect to find differences when I look more thoroughly. And so I just want to end by going back to what started this, which was looking at the distribution of these haplotypes in the wild. And so this is again the North Carolina population. I've rescaled the axis, but again, the northern and southern haplotype occur more frequently than you would expect. And I wanted to see if that varied with location along the cline. And so you can see in the north that the northern haplotype occurs even more frequently than in the central point in North Carolina. And in the south, the southern haplotype occurs extremely more frequently than you would expect. And so, in conclusion, we have these three naturally occurring polymorphisms, which appear to be clinal, that have non-additive performance with respect to fitness that's specific to adaptation in those different environments. This seems to be due to differential expression of the gene in which they are located, and it seems to be affecting their distribution of the haplotypes along the cline. So I just want to end by, again, thanking my collaborators on this project, Alan Bergland and Dmitri Petrov, as well as my advisor Paul Schmidt, other members of the Schmidt lab, our funding sources, and then other people who have helped out with the project. And thanking all of you for this wonderful first session, and enjoy the rest of the meeting. I have a few minutes for questions.
So you're attributing some of this variation. You're attributing some of this variation to couch potato, but in DGRP, I mean, what's the variation in all these life history traits that's present in DGRP? So potentially, like the variation you're seeing may be due to CPO, but it may also be due to all the variations present in DGRP.
Right. So the DGRP has a lot of variation in it in respect to these phenotypes, but by taking by creating these populations that are consistent only for these three alleles and letting them recombine, the only consistent difference among these different populations is these three SNPs in couch potato. So we let them recombine for about 10 generations, which should really mix up.
Oh, so these were all isogenic to those single SNPs?
Yes, so they were, I don't know if I can go back, but so essentially I took 9 lines that were, that had the three of the SNPs the same, and let them recombine for a while. So the background is really mixed. It's not, I'm not testing this on the individual DGRP lines, which you could then attribute a lot of differences to variation in the DGRP.
Not quite following.
Okay,
so the genetic background of these lines you're using are variable, or just those 3 SNPs are variable?
The background is variable. Within so each of the population cages, the SNPs are consistent.
Okay.
I let them recombine to the background.
So then potentially you can have background genetic effects that are actually affecting these life history traits, and not necessarily, those SNPs might be, but then there's also the genetic background there might be genes interacting with your gene, not just those alleles interacting with
There could be genes interacting with it, but because they're all of the other alleles will have a more heterogeneous background, to get the consistent patterns that we're seeing, I think you can contribute it to couch potato. The session's over, but feel free to, there's coffee break, so feel free to come up and talk more.